Okay, good morning, everybody. We'll get started here. I hope everybody can hear me and that you're seeing my screen. Um, looks like I'm working. So we left off Monday talking about some of these eukaryotes, and we were talking about a few of the ciliates. Let me get this to full screen real quick. So the previous one was paramecium, and we were looking at it a little bit. And you can see the oral groove right here, the mouth, if you will, of the parasite or of the ciliate. I oftentimes think of lots of these guys as parasites. Those guys are not. Stentor is another one, and it's really pretty impressive. These are really quite huge single-celled eukaryotes. Some of them, and I've got a video, I don't know how much of it I'm going to show you, but I'm going to show you a little of it here in a second. They describe some of them being upwards of a few, maybe 4 millimeters long, definitely visible with the naked eye. A lot of them we do look at through the microscope, and it's really definitely pretty impressive. So with that, you can see they have the potential with a holdfast to kind of anchor themselves to some substrate. They can elongate out their body in search of food stuff up at the top, what I'd like to call the head, but they don't really have a head. There's a bunch of cilia that are going to help to allow them to move, kind of, extend and contract, and also to help propel food into their mouth, the cytostome that we can see right here. So really pretty impressive. I believe Monday I mentioned this site. I'm going to see if you stop sharing that real fast. I'm going to see if I can get this other video to work at the same time. Let's see if I can share this. So this was a video from Journey to the Microcosmos. There are all sorts of videos that they have posted. So if you find this kind of interesting, I recommend you check it out. I will tell you the narrator, his voice is soothing. It's something that, yeah, definitely if you're looking to go to sleep, maybe he's someone that you want to listen to. So let's see if this will work. I believe I've got it on closed captions.
The most beloved of tiny organisms is probably the tardigrade. Bumbly, cute, chubby, and six-legged, they look like macro animals, hence their common names, moss piglet and water bear. And look, we love them too. We love them, and we will talk about them on this channel a great deal. But we also have a favorite species that you likely have never heard of. Larger than a tardigrade, though only a single cell. And the fact that you've probably never heard of Stentor is sad, because they are beautiful, magnificent, and powerful organisms. So, Stentor. It's a genus of single-celled eukaryote. There are 19 known species, but there are probably more than that. Some of them can build mucus houses that they can hang out in and hide inside of. Some carry algae inside of them that produce food for them. And all of them are astoundingly large for a single-celled organism. Some can be as much as 4 millimeters long, big enough to be seen without a microscope. The biggest tardigrades, which have thousands of cells, are 1.5 millimeters long. One thing every Stentor has in common is the holdfast organelle, which lets them anchor to a single location. Now, they can swim, but in an undisturbed culture, only a few stentors will be swimming freely. Stentors are actually more dense than water, which means that they sink. So that holdfast organelle lets them save energy once they find a location that has plenty of food and oxygen. But just because they're anchored, that doesn't mean they can't move. Most stentors are also capable of stretching a huge amount. When attached and feeding, a stentor body can reach 5 to 10 times their original length. And that's when it starts to resemble a trumpet, which is where it gets one of its common names, the trumpet animalcule. The biggest of all the stentors, and one of the largest single-celled organisms that exists, is Stentor caeruleus. They can be the length of a rice grain. There are insects in your backyard that are smaller than these massive but common unicellular organisms. They're in freshwater habitats all around the world, and we, here at Journey to the Microcosmos, have a pond where we find them all the time. Surprisingly, they thrive even when it's freezing cold. We have a stentor culture set from Stentor caeruleus collected under 20 centimeters of ice. Possibly the lack of predators during winter keeps their numbers extra high. Stentor caeruleus is a part of ciliates. Stentor caeruleus have hair-like structures called cilia. The beating of the cilia propels these cells in the water when they want to move around, and it also brings food particles, microanimals, and single-celled organisms into the cell mouth, where they are taken inside the cell to be digested.
That's what we were just seeing right there. This is a little paramecium that stentor was, well, it was trying to feed on, and that's what it's saying right up here. So this video, it's got 6 more minutes. We're going to stop it here. There are definitely a lot of cool things that they progress through and talk about with them, and numerous other, like I said, other videos that go through and talk about all sorts of things that they've found in their little pond and elsewhere. So if you are interested, you can seek this out. Let me flip back to our lecture. Okay, hopefully back in business. I think this is looks great. So the stentor, these ciliates, really very impressive, and as the video showed, a handful, 19 or more, at least 19 known, different species within this genera, and that there's all sorts of variation that we can see with them. Some of them harboring some photosynthetic algae inside, allowing for the ability to kind of make their own food, their sugars. Shifting over, this is a mycete. These are molds, and in this case, what we're looking at is, it looks rather moldy. It is covering, well, we can't really tell what sort of insect is under there, but there's an insect that this is in the process of degrading. So really pretty interesting. And, deep down, I bet you there's some of these on those videos as well. I just feel like they've got all sorts of cool things that they share. So Euglena, the Euglena itself, not something that we really think about in at least in introductory microbiology, because these things are not something that we think of as disease causing. They're definitely talked about in biology, and we use them to describe a little bit here. They are definitely microbes, and we don't want to just forget about them because they don't cause disease. But some of the others that are also within the same grouping do cause some serious disease. So just not photosynthetic like we're seeing over here. So these guys have the pellicle that allows them to basically kind of have like a sort of an internal scaffolding, like a cytoskeleton that confers some of their shape. Again, with the euglena full of some chloroplasts, the ones we're going to think about here in a second, not. These guys also have an eye spot that allows them to sense sunlight, thinking of them like in a pond or some aquatic situation, kind of knowing where they're at in the water table, moving toward the top when it's daylight and they could photosynthesize, and then as the sun goes down, maybe being at the surface of the water isn't the best, so they recognize that and can maybe sink down a little bit. So pretty cool. Now shifting over to something maybe a little more disturbing, definitely. The trypanosomes, these again fit within the euglena grouping. Trypanosomes can cause some serious issues, potential fatal issues with infection. So this again is kind of just these first chapters in the text are really kind of introductory to just introduce a few of these things. Although we're looking at a life cycle here, there are more details with trypanosomes a little later on when we start thinking about some of the infections, specifically thinking about blood infections, because these are where we would find them. We call them hemoflagellates because they are flagellated parasites within the circulation. So again, they're, like I said, kind of glossing some stuff over initially. Trypanosoma brucei is the genus, but truly when we think about these guys, there are some subspecies, so we further divide them. If we were to look at them morphologically, you couldn't tell them apart, but they are different. They're different as far as the type of disease that they might cause. They're different as far as what they might infect. There are 3 different types of them, one of them only infecting livestock and 2 of them infecting people. So I'm not going to bog you down with that stuff. We'll just focus on the trypanosomes just as a generalization. It causes African trypanosomiasis. We also call this African sleeping sickness. And again, like I said, really two different ones that impact humans. And the severity of the infection is definitely different with that. Some of it is very acute, meaning it targets you very rapidly, and prognosis super poor unless you can receive treatment. So these have 2 hosts for the life cycle. The tsetse fly right here is what is going to transmit it to humans as it takes a blood meal. It injects the parasite with its saliva into us. And there are a handful of different types of developmental stages. Again, we're not going to go through that, but a stage within the human that's infective to the fly, multiple stages of development inside of the fly, moving up into the salivary gland of the fly, and becoming the infective stage to us. As that fly takes, the tsetse fly takes a blood meal with the saliva, it injects that stage, and then they get in to our circulation, they start replicating, and well, definitely causing some serious issues. Again, species specific as far as what or whom they may be targeting. And images, I think we saw an image before with some of these trypanosomes in circulation, so red cells and them together, and they really are quite striking to see them in a blood smear. So I'm going to back it up here real quick. There's another one that's grouped up similar with the trypanosomes that we call the leishmania. And although they're not transmitted by a tsetse fly, it's a sandfly. They have a similar cycle infecting within our circulation and lymphatics, different courses of infection, definitely. And we'll touch on a little of that later on as well. So shifting over slightly, or maybe a lot, we now want to think some more details about the helminths. So moving up to all sorts of different types of worms, multicellular, and in some instances potentially quite large. It kind of depends on the circumstances. And again, really kind of just some introductory stuff as we start moving through here. There are more details in the latter chapters that describe some of the actual infection. So this one that we're starting with to introduce the worms, this is a type of nematode, so we call these guys roundworms. This little guy is called Enterobius vermicularis. It's commonly known as a pinworm. It's also been called the seat worm. And you know, I like to think of it as fun for the whole family. This worm is something that is really pretty common. It's something that's pretty easily transmitted, and the infection that it causes is overall pretty mild. Maybe individuals could be asymptomatic, not recognizing they've even got any issues. Little kids can have some trouble for sure. Restlessness, irritability, things like that can cause some problems. So it kind of just depends. I'm looking at a chat right now that popped up and it says dogs get roundworm as well. Correct. That is correct. And how does this spread to them or vice versa? That is a great question. And let me close this off. There are all sorts of potentials for transmission of worms, and again, dependent upon the types of worms. If we're talking, you know, like roundworms, oftentimes we think about the eggs, and with pinworms, that's what we think about. The eggs are what's infective, so they get ingested. You know, a contamination issue or something like that. Fecal oral contamination sounds very gross, but these eggs are spread and shed and contaminate all sorts of things. They get in dust particles and can basically move throughout the house. We think more like clothing and bedding and things like that, but literally they can be found everywhere, which is also disturbing. So things like that, you know, coming in contact with the egg and accidentally ingesting it, you don't see them, they're microscopic. And in turn, that's how you can become infected. With dogs and roundworms, there's all sorts of potentials, again, for the types of roundworms. But oftentimes it's something like that. So eating something that has the eggs on it, or eating you know, a rodent or something like that that has the parasites. And typically in those circumstances, in the wild animal, in the rodent, it's in a cyst stage. So in the musculature typically of that animal, there's a cyst with the parasite basically hiding or waiting underneath. When it's consumed by an animal, it's able to exist, so kind of hatch out, and that in turn lends to infection. Again, tremendous variation with the types of worms and the body sites that they might be infecting. So hopefully that answered your question. We'll see a couple more examples of these worms, not the pinworms, but we will talk pinworm a little bit later on. So it is, you know, it's like I said, it's something that's, you know, as far as symptoms go, very, very mild. Typically, it doesn't cause serious issues overall, except, you know, it's a little bit disturbing. And if you know, usually it's little kids that we start thinking about that might have this. But I said fun for the whole family because it is really pretty contagious, as the eggs are literally spread all over the place, it sets the stage for problems. So we'll skip over the rest of my details with pinworm. I feel like I could talk about pinworms for a whole hour, but I won't. I will spare you of that. So there are a lot of different types of helminths, and the pinworm that we were just looking at, I mentioned, was a nematode. We call those guys roundworms. These ones that we're looking at are a little bit different. These are the flatworms, so platyhelminthes, and we can divide them into some different classes. The turbularians up here at the top are free living. We don't think of them as being parasitic. They're kind of oddballs. There's some variation within them, and we're not going to go into great detail with that. The Monogeneida, these guys are types of worms that infect a single host. That's what this is really meaning with that. And in this case, lots of times we think fish. Dactylogyrus is a common one that gets into the gill filaments of them. The next two, the Trematoda and Cestoda, we call these guys digynians, typically two hosts, maybe more, but usually two hosts that participate in this. And with a lot of the flukes that we're looking at right here, the trematodes, oftentimes we see a snail, either an aquatic or a land snail, serving as an intermediary host, and then maybe us or some other vertebrate serving as a definitive host. With the cestodes, these are the tapeworms. We usually think of, different types of animals. So us and maybe other types of livestock, pigs, cattle, even fish, with some potentials for some of these really surprisingly huge worms, as we see this ribbon-like worm. And this is Taenius agenata, which is the beef tapeworm, potentially meters long, 4 to 10, we say here. And it kind of also depends on circumstances or location, I should say. In the US, they typically are on the smaller scale, although 4 meters seems disturbingly large. Elsewhere, they typically are much larger, 10 or maybe even approaching upwards of 20 meters long. And that, well, I like to say that's truly a buttload of worms. Okay. So again, lots of potentials. Really, it's these two down here, the trematodes and the cestodes, that we think as far as disease causing, the fluke and the tapeworm, respectively. With the trematodes, these guys typically kind of flattened, dorsoventrally flattened. They're sort of leaf-shaped, and definitely vary in size. Nowhere near thinking the tapeworm magnitude. They're definitely not meters long. All right, and with both of these trematodes and cestodes, for the most part, we think of these guys being hermaphroditic. That would be monoecious, so both sexes harbored within the one worm. There are a few exceptions with some of the trematodes. As far as cestodes go, I can't think of a single cestode that's not hermaphroditic. And each one of the segments, although we're not seeing it, there's thousands of segments on this worm. Each one of them harbors the reproductive structures. Individual proglottid gets full of eggs, which either ruptures, releasing eggs out, or the proglottid itself starts to break off, and those are defecated out. And in turn, contamination sets the stage for something to consume it, and the cycle continues. So it typically, as we think of these things, like the tenias here, the Tinea saginata, we get infected with that by consuming beef that is raw or very undercooked, that allows the cyst within the musculature of that animal to not be destroyed. And as it's consumed, it allows it to exist. And there you go. You're infected. The adult stage of the worm results in your intestine. And that's what we're seeing right here. So in the cow, it's serving as an intermediate host. Within the musculature is where the parasite is found. You don't see it in its intestine, robbing it of nutrients. It eats the egg, and the larva insists in the musculature. We ingest the larva, and we get the adult stage of the worm. There are a couple that can cause some other issues. Taenia solium is the pork tapeworm, and we get infected similarly. Eating pork that is poorly cooked allows that cyst to be ingested. But with Taenia solium, it also holds another issue because if through poor hygiene, if the eggs are ingested, so you use the restroom, you don't wash your hands properly, you end up consuming some of those eggs. Now, in this case with Taenia solium, you can serve as an intermediate host as well as the definitive host. So in your musculature, the larva will insist, maybe even go into your brain. But if you've ingested the larva, you also have the worms in your intestine. So it's you're serving as both intermediate and definitive host. There's another roundworm, Trichinella, that also does similar things, where the adult stage of the worm in the intestine, but through egg ingestion, not larva in the meat, egg ingestion causes it to insist within our body. So kind of crazy. And I'm pretty sure it was the Trichinella. The text kind of mentions it in some detail, but it's also a little bit misleading because it says, consumption of poorly cooked meat can result in those parasites insisting in your musculature, and that's not the case. Consumption of poorly cooked meat allows you to get the worm. The worm lives in your intestine, and then it's through contamination issues where you ingest the egg, not the poorly cooked meat, that allows it to insist in your musculature. So I wanted to point that out. If you're actually reading the text, which I hope you are, there's a little bit there that's kind of overgeneralized, I felt like. So here we've got a couple. Epistholorchis viverni is both of these worms. This is a fluke. And the next one over, although we're only seeing the head region, the scolex, this is a tapeworm. So they're both flatworms, but fluke and so trematode and cestode respectively. Lots of these, both trematodes and cestodes, have oral suckers. They don't have all of the features like we think about, like ourselves, with a complete digestive tract and all sorts of complexities. They're still complex, but they're parasitic and they have very reduced body plans. Oftentimes, they may have a mouth that goes right to a blind sac, or none of that at all. They absorb nutrients directly through their tegument, so skin. Both of these guys can do that. The roundworms have a complete digestive tract, mouth to anus, and they pull in food through their mouth and digest it, and they don't absorb directly across they don't have tegument, they have a cuticle, right? So the Taenia solium, again, this is the pork tapeworm. It's got suckers, and it also has some ornamentation. It's got some hooks up here that allow it to attach to the intestinal epithelial cells, and the little barbs, the little hooks, take hold within there. The suckers also help them to attach. They're not sucking in food. It's just a hold fast. It's just a mechanism to allow them to adhere. If we could see below the scolex region, you would see the individual proglottids, the segments found within these guys. And they're a little bit smaller than the Taenia saginata that we were looking at, upwards of 4 meters long. They're a little bit smaller than that, but still definitely impressive size. These next ones, these are a type of fluke, and they are ones that are different in the fact that they are dioecious, so they are separate sexes. Dioecious means two houses, so male and female separate. And really, as far as thinking parasites, or the worms, I should say, they're really kind of an exception for that. Like I said a second ago, most of them are hermaphroditic. So these guys are super impressive. There's all sorts of potentials with this, and there are a few here that we can see that cause some serious disease as they get into our body and truly migrate where they need to go and complete their life cycle, releasing eggs for the cycle to continue. Fortunately, around here, we don't see these guys. You have to travel abroad to come in contact with this. South America, maybe, Africa for sure, and moving into Asia, parts of Asia, you definitely can pick up some of this as well, with Japonicum as an example. Around here, there are some potentials to get schistosomes, but we're not the right host, so they can't complete their migration to typically GI tract, where they want to go. They can't. They stay localized in the skin, and what we end up getting, we call swimmer's itch. It's something that is pretty stinking common in the summer months. You definitely hear stories of it around here. There's a few lakes that I know have had issues. Head up north, Rigby Lake, definitely issues in the past. Jensen's Grove and Blackfoot, also potentials. And I'm sure there are other locations. Those are two off the top of my head that I have heard of. So definitely, yep. Someone say, I got it from Blackfoot. It's horrible. That's what I've heard. So Jensen's Grove, I'm assuming. And yeah, it's, let me describe it a little bit, and I'm sure you will say, yep, that's true. These guys, the Cercaria, these are the little juveniles. They want to get in and penetrate into the skin. That's what this little eye is right here saying. That's what's infective. All right. Well, before we get to that, the eggs released out into the water by us or by some animal, if we're thinking like Jensen's Grove, likely from a duck, because aquatic fowl are really what this would want to be infecting in the first place. But we happen to be in the wrong place at the right time. So either secreted in the feces or in the urine. Hematobium is one that we see within the urine, and so within the bladder, there are definitely some issues with that. They have some spines, little spikes on them. Hematobium can cause some serious issues with the lining of the bladder and potentially lend to bladder cancer. And we can kind of diagnose based off of the way these eggs look, spines or not. So the egg is released in the feces or in the urine, dependent. If it makes its way to the water, they are able to hatch. When they hatch, they have a little ciliated organism that's inside of this. It looks like a type of protist, but it's not. It's a worm. It's got the cilia that allows it to swim around. It senses the mucus from a snail. It smells that and swims toward it, and it penetrates in using kind of an augury motion. It burrows into the snail, and in the process of doing that, it sheds that epithelial cell, loses the cilia, and becomes a sporocyst inside of the snail. The sporocyst grows and develops, and it generates maybe multiple types of intermediaries within it. We finally produce these cercaria that burst out of the sporocyst, are released out of the snail, either penetrating through its tissues or released in the mucus of the snail. There's a couple of different possibilities. These cercaria are flagellated, as you can see right here, little forked tail. They swim around very rapidly. And if they sense, just like the Miracidia sensing a snail, if they sense a host, they will penetrate through the skin and try to develop. If it happens to be these guys, we're the proper host for that, and they migrate either into the hepatic portal veins or the veins of the bladder plexus. Either way, taking up residence. And there you go. First off, they wind up within the liver, and that's where the male and female find one another. They pair up, they start to mate, and well, they pair up and then they start migrating to their final spot within the GI tract, like I said, or bladder. Once they're paired up, they basically, well, the male has a gynecophoral groove, so it's kind of a split body. The female swims up into that, and he closes himself up around her. They pair up, they're locked together forever. Basically in a bout of constant copulation, we could describe it. Constantly breeding and pumping out eggs for their entirety. That is crazy. And in turn, hoping to make their way to the water for the cycle to be complete. So the stuff we see around here, typically, like I said, is something that wants to get into a bird, not into us. But we happen to get into the water. The cercaria there, they only survive for a couple of days. If they don't penetrate into a host, all of their energy is gone, and that's the end. Whatever. They penetrate through our tissue or get right into our skin, but these guys, like I said, wanting to get the birds, can't migrate like they should, so they stay localized within the skin. It causes some really, I said swimmers itch, really intense itching. It almost kind of looks hive-like, welts all over where, basically each little bump that's raised up is the result of one of these cercaria penetrating in. You get a localized reaction within the skin as our cells try to destroy it and basically mop up all of the issues. So it's, as whoever said a minute ago, it sucked. So not good. Okay, moving on from the schistosomes. This, again, definitely some potentials. I have a sister-in-law that took a trip to Africa maybe two years ago, maybe a little bit more than that. She went with a friend, and I told her a little bit about some of the issues before she had traveled, and I definitely told her about some schistosomes when she said some of the places she was going. And one of them, Lake Malawi, is known for its issues with schistosomes. And people go there, they get in the water, and unknowingly they contract it. She did not get into the water, but her friend did. Maybe she didn't listen to her warnings. I don't know. But she got in. Her friend later returned back to the States, and if I remember right, was in Alaska, working in Alaska, and definitely not on the radar up there as she started contracting these issues with it, and it took quite a long time to get the people to really start thinking this is what's going on. So crazy. Africa was a place that I was like, that's on my bucket list. I want to go there. And thinking about all of the possibilities of parasitism, now I'm like, maybe just looking at some pictures is okay. That's all right. We don't know. All right. We've already kind of hit these guys the littlest bit, but I know you want to keep thinking about worms. Why would you not? Um, so here we can see a couple of worms and, the saginata or the solium. And again, the life cycle, the way that they infect and issues, pretty similar. Uh, whom we're getting it from, a little different. All right. So we release out proglottids. Here you can see the individual proglottid. Here's actually a couple of them. And inside, we've got all of the reproductive structures. A lot of this dark stuff that we're seeing here are eggs and what we call vitellaria, which is cells that provide like the nourishment, the yolk for that egg. So each of these proglottids is full of all of that to ensure that all of the eggs get everything they need. You can see a lot of it right within here. Again, more reproductive structure, eggs and vitellaria. So this is what's released out of us. We say here diagnostic. So, fecal specimen with proglottids or with the eggs within it, dependent upon the circumstances, can give us diagnosis. In some circumstances, it's the defecation of a chunk of proglottid that's your first indication that you got worms. That's maybe a little bit disturbing. So as these are passed out into the environment, the animals, the livestock get infected when they ingest vegetation contaminated with that. So, in the United States, we hope maybe that this potential is less. If we're thinking about us harboring it, really that means these guys need to be eating our excrement. And if we can drive a wedge in that, we potentially prevent them from contracting it and the whole cycle. So we do see tapeworm in the US, but incidence is considerably lower than if you looked abroad, right? So definitely some issues with that. I read a little snippet of, I can't remember where I actually saw this. It might even have been in our text, talking about saginata and the cows in India. These are holy, and they kind of roam all over the place. And there are definite circumstances where someone, trucks off into the brush or the forest to, do their business, find a tree and there you go. And the cow follows them and waits till they're done, and then it consumes what they've left, and that is disturbing. So, whatever. But we hope we can drive this wedge. If they do consume that, they ingest those eggs, the eggs hatch, they move to the musculature and encyst. So that's what we're looking at right here. These oncospheres are juvenile stage of the worm, an intermediary, and that's what the livestock is serving as, is an intermediate host. So within the musculature, these oncospheres develop into the cysticerci, which are what's ultimately infective to us if we consume raw or undercooked meat. We can get it. So saginata, solium, and they attach into the intestine and grow. There's another that we could have over here as well. It's not the tenias that we're looking at, but Diphyllobothrium latum, and you don't need to get bogged down with all of that. But Diphyllobothrium is the broadfish tapeworm. So the consumption of raw fish, sushi, may set the stage for you ingesting similar idea and getting a large worm within your intestines. Kind of depends on the circumstances. It depends on the type of sushi. There are numerous things, and if it's treated properly, you would likely have no issues. So I don't want to turn you away from eating sushi because it's delicious. This next one I think is pretty impressive. It's a little bit disturbing at the same time. This guinea worm, guinea worm is a roundworm, a nematode. Dracunculus is the genus. Dracunculus mediensis is the guinea worm, and the infection we call dracunculiasis. This is something that it's impressive. It's something that we are on the cusp of saying we have it eradicated. There are very few cases of it now. There are only a couple of locations where we still see it endemic. A couple decades back, there were thousands of cases of this, and now very, very low. So we hope that this is one that we actually are able to truly deal with and eradicate. We get infected by typically drinking water that is contaminated. Within that water is a type of organism. We call it a copepod, but really that's just a developmental stage of an invertebrate. So typically maybe a type of juvenile arthropod, something like that. I've heard them described, oftentimes called water fleas. And again, it's some larval stage of something else, but they become infected, and their accidental consumption, you typically don't even recognize it in the water. As it's consumed, the worm ultimately infects us. So pretty interesting, and I know we'll mention a little more of it later, but these guys, the female or the worm, after eggs are fertilized within her, and I'm almost, well, with the nematodes, they are dioecious, separate sexes. So yes, male and female. The female gets her eggs fertilized, and after, and can be upwards of about 1 meter long, migrates down, and this looks likely on the ankle. It's almost always on the foot, right? They move down to the extremities. An ulcer forms usually in something like this, pretty painful. You want to put your foot in some water, which is exactly what that worm wants, and eggs are released out into the water. Cycle hopefully being completed. You can, as this worm is emerging, as we see here, a little matchstick, you can kind of twist that worm out. It takes days, potentially weeks, of slowly, kind of methodically just turning the matchstick to slowly pull it out. You don't want to break the worm off inside, or you're going to get a pretty nasty reaction to that. But it's still a common treatment to remove these worms like that, which is pretty crazy. So how do you get the worm out of the skin and onto the matchstick? Well, ultimately the worm emerges hoping to lay the eggs. So, soaking your foot in some water can get it to kind of protrude out through, you got to wait till the skin ulcerates in the first place, because then it's ready to emerge and do that. And so that will help entice it, soaking your foot in some water or something like that, entices it to come out, and then you can slowly turn it out. And there's, ancient like wood cuttings and things like that that show this, these doctors, if you will, removing worms from people's feet. Pretty cool. I got another question on here. We are right at the end of the lecture. Are all of the exams going to be proctored? And the answer to that is yes. They're going to be proctored. And is the next one going to focus on it's going to be 4, 5, and 6. So one more chapter. We will wrap this chapter up Friday, and then we will focus on chapter 6 next week. I will try in the meantime, before next week, to get a summary and a study guide posted for you guys. So you've got something to review and some kind of guided questions that will help out with that. Any other questions? Okay, so Friday we will pick up with the different types of fungi, and that's the end of this chunk of material, or the fungi. And there are lots of, some details within here that I truly, I don't want you to get bogged down with some of that. Okay, if there are no other questions, I hope you all have a great day. Farewell.